Vanakkam. Welcome to this course on classics in neuroscience. In this set of videos, we have been looking at cell biology and the synapse and how the synapse came to be first identified and described. In this video, we will be looking at the contributions of Rene Couteaux. Do not know the exact pronunciation, but do check, and George Koelle and his contributions. An example of how difficult it was for researchers of that age. Rene Couteaux actually performed some of his experiments under Nazi occupation in the 1940s in France. Remember, in 1940s, France was occupied by Nazi Germany, by Hitler's forces. Rene Couteaux actually performed his research under this occupation. The other story is, of course, of the story of Bernard Katz. I requested you to read about that. Please do check about the story of Bernard Katz and how he escaped from Nazi Germany and went on to live with A.V. Hill, and essentially in the lab of A.V. Hill, and later went on to become a Nobel laureate, established his own fantastic lab in Australia and so on and so forth. Do read about the difficulties that these researchers had. Compare them with the difficulties that we have. What are our biggest complaints? That this equipment is not turning on, or there is some small issue with this calibration of this equipment. Comparatively silly when compared with life-threatening consequences that these individuals had. Anyhow, stories apart, let us move on to this. So, let us look at Rene Couteaux and his contributions. Rene Couteaux was a French medical professional or a doctor, physician, and his aim was to study and understand the structures of the neuromuscular junction. He essentially questioned, is the neuromuscular junction separate? That is there a neuron and a muscle, or are these fused or continuous, supporting the reticular theory? This was an age-old question, but he started studying this only in the 1910s, so it was not that old at that time. Several methods were used, such as gold and silver impregnation and vital staining with methylene blue. Several of these methods were used. These methods did not provide clear visualization of neural and muscle membranes and did not show whether they were indeed separate. By mid 1930s, it was already established and accepted that acetylcholine was a neurotransmitter. Actually, adrenaline was also shown previously as one of the neurotransmitters. So, acetylcholine was established as a neurotransmitter. So, Couteaux, he changed his focus from acetylcholine to acetylcholine esterase, because the description of acetylcholine required that after its use in the neuromuscular junction, there must be a way to remove the used acetylcholine from the junction so that the next muscle contraction can happen. This was the requirement. A question is, how did acetylcholine get itself removed? How was it removed from the neuromuscular junction, or the cleft or the gap between the neuron and the muscle fiber? It was later suggested that this was perhaps made possible through the enzyme acetylcholine esterase, ACHE, acetylcholine esterase. Rene Couteaux proposed that acetylcholine esterase perhaps played a crucial role in inactivating or in cleaving, in hydrolyzing acetylcholine after acetylcholine does its job on the postsynaptic muscle fiber. This work he did in collaboration with David Necmonson, who was a German Jew who fled from Nazi Germany to France, and he worked with Rene Couteaux. They used biochemical methods to identify and study acetylcholine esterase, and they found that indeed in the gap between the neurons and muscles, or the sites where the neurons innervate the muscles, or the neuromuscular junction, acetylcholine esterase was found. It was localized and it persisted even after the degeneration of the nerves. This suggested that acetylcholine esterase is present in the neuromuscular junction themselves, in the membrane itself. Further, during World War II, under very difficult conditions, Rene Couteaux used a dye called Janus green B to reveal a synaptic gutter, a sort of a gap at the neuromuscular junction with some sort of subneural apparatus on the muscle side. This provided strong support to the neuron doctrine, essentially suggesting a separation, a gap between the neuron and the muscle. For the first time, there was a clear morphological evidence to suggest that there was indeed a separation between a neuron and a muscle, and there was a chemical agent, which is acetylcholine, that was responsible for activating the muscle or muscle contraction as soon as it is released from the neuron, and this acetylcholine was destroyed or hydrolyzed by the enzyme acetylcholine esterase. So, this essentially led the foundational groundwork for improving our understanding of synapses or synaptic structures and functions. His findings to a great extent influenced the neuron doctrine and the broader field of neuroscience. Unfortunately, it did not get the credit that it properly deserved because the work was published in French, a language other than English. Remember, English is the language of science as of now. Because this work was perhaps published in French, it somehow did not become visible to the many researchers on the two sides of the Atlantic who are working in this field, because those researchers working on the two sides of the Atlantic, that is in Britain and in the United States, they are essentially publishing in English. Although there was biochemical demonstration of localization of acetylcholine esterase by Rene Couteaux and Neckmanson, it was still not convincing enough for some of the people who were doubting, who were skeptical. That is when George Koelle comes into the picture. George Koelle was born in 1918 and he studied at Swarthmore College, and he received his PhD from University of Pennsylvania in 1946. He essentially studied the need for localization, very precise localization of acetylcholine esterase, and he used a relatively novel method, and his method was a histochemical method using a closely related molecule, a molecule that is closely related to acetylcholine esterase, that is acetylthiocholine. It turns out that visualization of acetylthiocholine is easier when compared with visualization of acetylcholine esterase. So, he used acetylthiocholine for staining, and this method for the first time showed localization of acetylcholine esterase. This work he published with Friedenwald in 1949. This method came to be used widely well within the central nervous system, and this method came to be called as the Koelle method to identify and demonstrate cholinergic systems all over the central nervous system, and this method was used everywhere. Koelle continued to be completely devoted to this line of research well into the 1980s and in 1990s. He was always focused on this topic. Rene Couteaux and his team improved on this basic method proposed by George Koelle to provide more evidence for the existence and localization of acetylcholine esterase. With this, we come to the end of this video. In this video, we looked at the contributions of Rene Couteaux in trying circumstances of the World War, and George Koelle and his contribution. Koelle with 2 L's. Okay. Thank you very much for your attention.